Ancient Human Genome Sequence of an Extinct Paleo-Eskimo, a presentation by Group A29, Megan White, Kelsey Sargis, Kate Nickerson, and Camilla Dartnell. Holy DNA!
What's going on?
Well, this ancient Paleo-Eskimo hair from about 4,000 years ago was brought in, and we were able to break it down into genetic components to find out so much about life back then. Step into my office so I can show you all the details.
A team set out to excavate in Greenland. When they came to the specific region, they discovered 4 ancient hair tufts. After further examination and testing, they found out that they were over 4,000 years old and well preserved. Knowing this information, the team wanted to further investigate and find out the origin of these hairs. Because they traveled to labs in Copenhagen, they had to check for contamination of the hairs from the ride. It was able to be determined which was the most preserved and least damaged through a series of tests. Even though the hairs were well preserved, the team needed to check for contamination caused after the excavation, which could occur when someone handles the hairs or while they are transported from the site to the labs. Luckily, the team used great protocol to try their best to prevent this. If contamination did occur, we could spot it easily because it would be Northern European, the ethnicity of anyone who could have handled the hairs at some point. The hair that we used to find all of our data had less than 0.8 contamination. This is truly remarkable considering the age of the hair.
One of the important discoveries that we made has to do with the population that the ancient human was part of. Our specimen was part of the Saskek population, which is a Paleo-Eskimo culture population. Past theories linked the existence of these populations with the Native American populations, claiming that the Native American populations originated from the Paleo-Eskimo populations. A Saskek genome analysis found that this Saskek population, in fact, was separate from the Native American populations, finding that the people of the Saskek tribe did not contain any distinctive genotypes to either the South or Central American populations. The most related influences to the Saskek, according to the data gathered from our individual, are the native East Asians and the populations of what we now know as the Asian and North American sides of the Bering Strait in the Arctic. This means that the Saskek population probably left their original population in Siberia just prior to migrating to the area where the hair specimen was found, instead of mixing with many other populations for generations before their relocation. A really cool thing we were able to find out from just the mitochondria DNA, or mtDNA, was specific phenotypes or physical traits and characteristics of the human being. We deducted that he had brown eyes, dark, thick hair, with a tendency of baldness, dry earwax, and teeth, with characteristics specific to populations from Asia. We were also able to find out that his blood type was A positive, and that his body's metabolism was well adapted to a cold climate.
By analyzing the single nucleotide polymorphisms, aka SNPs or SNPs, on the DNA sequence from the hair and comparing them to common SNPs of different races and religions, we could determine all the traits found about the Saskek individual. There were 350,000 SNPs that matched. This shows that there is a 99% chance that the phenotypes that we predict he had were actually true. Now you're probably still confused as to what a SNP is. A SNP is a single piece of someone's DNA that usually differs in individuals. Since these genetic variations are common with different people, scientists identify them on unknown DNA to figure out whose DNA it most closely represents. For example, a combination of 4 different SNPs on a certain region of a chromosome, for our purposes, you don't need to know exactly where, looks just like ones found among Asians with brown eyes. Therefore, we can conclude that the Saskek individual has brown eyes. See how simple that is? It's all compare and contrast, just on the molecular level.
I can't contain my excitement about this finding. Before this, 0.7 times was the most in-depth an ancient nuclear genome had been sequenced. Now we've successfully sequenced this ancient human's genome to about 20 times, which is over 28 times what we were able to see before. Also, we were able to sequence 80% of the entire genome, which is remarkable considering the age of the human.
So let me get this straight. You were able to find out what they looked like, where they originated from?
Yes,